Hello, I'm Bradley Alicea. I'm the person who heads up the DevoWorm group. And a few weeks ago, uh, Dr. Richard Gordon died. He passed away of blood cancer. And it was a very sudden, he had not been in good health for a while, but it was a really sudden event. So he died on June 14th at age 82. And this is a shot of his Google Scholar page. He's a very accomplished academic. He was a family man, and he went by many titles. Two of the ones that I was involved with most were his role as sort of a theoretical biologist and an eclectic biologist. So he was a theoretical biologist, but he was also an interdisciplinary theoretician. So he was interested in a wide range of biological questions, from biophysics to, as we'll see, cancer physics, to other types of biological questions in embryology and in evolution and other areas as well. That's where the phrase eclectic biologist comes from. Usually in biology, we study things in terms of their domain. So if you're a developmental biologist or an evolutionary biologist or a cell biologist, you usually study that thing, and which can lead, of course, to a lot of blind spots. Dr. Gordon was very transdisciplinary in that way. He addressed problems with all different types of perspectives, and so he was a very good, broad, wide-ranging thinker. And so we'll go over some of the work that I've done with him, some of the work from his Google Scholar, and of course, his legacy. So I went to Google Scholar and I analyzed his most cited publications. And by most cited publications, I mean anything that got 10 citations or more. So one thing about Google Scholar is it's confusing him with another Richard Gordon. So I had to sort those works out. So if you go to his Google Scholar and you see a lot of publications on the history and philosophy of religion, those are not his publications. But he did publish in a wide range of fields. So this is all of the publications with 10 or more citations. And as you can see on the right-hand side of the slide, we have, his most cited paper was published in 1970, and it has about 4,200 citations. If we go down the list, we see that he has many citations in the hundreds, or many publications in the hundreds of citations. And so, and they range across a number of fields, and they were published from about 1970 to right before he died. So if we look at the share of different areas of his publications, it's quite interesting. So I said that he was, his perspective was wide-ranging, but he had some areas that he specialized in nonetheless. So one area he was very interested in was early life. And he published 12 papers in the early life area that had gotten 10 or more citations. Then he published 31 papers in the areas of computational biology and modeling. In fact, that's how we met, was through this interest, or the shared interest in computational biology and biological modeling. Then he has 39 publications in the area of diatoms and nanotechnology. So he was a true believer in the potential of diatoms as a model organism. 49 publications with over 10 citations in the area of embryogenesis, which is actually kind of his signature area, I think. That's what he had his lab on at the University of Manitoba. But he had this, he had these other interests that actually outstripped that, initial interest in embryogenesis, and then 48 publications in cancer physics, which he devoted his last years to. And it's not necessarily because he had cancer, but because he was interested in solving breast cancer, solving other types of cancer. So he was very devoted to that area towards the end of his life. And when we talk about cancer physics, we're talking about imaging technologies. We're talking about different types of computational techniques that make it easier to study and diagnose cancers. So this is something that he published back in 2010. It was from a blog of his that he had for a while and kind of gave up on, but which was called The Cosmic Embryo. And if you're familiar with the Erdos number, then you'll kind of appreciate this joke. So the Erdos number it's kind of like the 6 degrees of Kevin Bacon. And that is that Paul Erdos was a very prolific collaborator. He's a mathematician. And he would collaborate with all sorts of people in mathematics, but also outside of mathematics. And the idea was that he collaborated with so many people that you should have some sort of Erdos number if you're a scientist or if you're a published scientist and you've published with collaborators, that you should have some sort of Erdos number, and it's kind of similar to the 6 degrees of Kevin Bacon. So a lot of people have an Erdos number of 3 or 4 or 5. And I can't remember what mine is. I think it's like 2. But his Erdos number is 2I, which is supposedly an imaginary number. Now, this is a joke because that's not how we calculate the Erdos number. But in any case, he calculated this in a kind of an interesting way. So after receiving his PhD, he did a postdoc with Stanislaw Mulam, who is a physicist mathematician. And his Erdos number was 1, so he published directly with Paul Erdos. And he never published with Stanislaw because they didn't really see eye to eye on a lot of things. But he did a postdoc with him, and so his Erdos number was 2I. So it's a 2 an imaginary number. So it's, I guess, whatever that would be relative to 1. So that was an interesting kind of take on that. So I met Richard Gordon in the embryo physics course that he taught in Second Life. So if people remember Second Life, I think Second Life is still around. It was this virtual world. You would log in on your laptop. You would go into some space, and they have these servers that represent sort of physical space or virtual space that represent physical space. And people could get real estate in Second Life and build little communities. And so him and his wife, Natalie, they built this place called Silverbog. And in Silverbog, they hosted the embryo physics course. And so you can see here that I have my avatar here. And they had some like virtual whiteboards where they would project the talks. They would have a little virtual area where your avatar could sit down and enjoy the talk, and then you could ask questions in the talk, and it was all a very nice way to bring people together from around the world and have them give talks to one another and collaborate. I presented in the embryo physics course twice. I don't have recordings of those, but it was a nice experience. Now, I was at Michigan State University during the early 2010s, and I was invited Richard to give a talk to a group of people there. And this is a talk that he gave. It's called Cause and Effect in the Interaction Between Embryogenesis and the Genome. So at the time I was working in the area of cellular reprogramming, and that was, of course, very heavily molecular and cellular enterprise. And so this was, I think, a suitable talk for the lab that I was in, plus some of the people in our area who, did things like development and other types of biotechnology and bioengineering. So it was quite a nice, an appropriate talk for that. So he came to Michigan and he visited me on campus. And, he would do that. He and his wife traveled during the course of the year. They traveled to visit collaborators. They had like a fifth wheel, which is a type of camper, and they drive around to different collaborators, stay at campgrounds, and then visit them either at a university or at at their house or wherever they were. So he gave this talk in 2013. It was, I think, well received, and yeah, so it was interesting. That's kind of we had collaborated online, and then I finally met him in person in 2013. Then I moved to Champaign Urbana, Illinois, to the University of Illinois in 2014. And so he came to visit me in 2014. And he gave a talk at the Champaign Public Library, which is a very nice public library. Um, and so that was, we rented a room and we had a small talk there. And so in 2014, we also decided to start this group called DevoWorm. Now, DevoWorm has its roots in OpenWorm and the OpenWorm Foundation. So you can see Stephen Larson here, who was one of the people who started OpenWorm. And he was interested in having this developmental arm of OpenWorm, where we, would look at the development of C. elegans. And so this was something that we, we got together on April 11th. We were all in a Google Hangout, if you remember what those were. They're a very early version of, um, teleconference software. And then we hashed out some details for first steps. So this was in April of 2014. So this is over 10 years ago. And we've kind of been going with DevoWorm ever since. We had weekly meetings from about that time, and we would invite people to talk, or we'd have very informal meetings where we'd kind of work out problems. Interestingly, we have a talk on we have a talk on YouTube. The talk is on the 10th anniversary of DevoWorm, and I go over sort of the early years of DevoWorm. One of the earliest parts of DevoWorm was where the initial sort of impetus for DevoWorm was this issue that was on the OpenWorm GitHub issue board. And it was about create a model of the embryo, and kind of outstripped that original issue. But we've had a very fruitful collaboration, a very fruitful set of activities as a group. So in 1999, Dick published this book called The Hierarchical Genome and Differentiation Waves. And this was the culmination of a lot of the theoretical ideas he had while working as a developmental biologist. So he had this idea that there were these differentiation waves in development, and that those could be translated into differentiation trees, and then furthermore in a differentiation code. And I call this jokingly this white whale. So if you're familiar with the book Moby Dick, in Moby Dick, the captain of the ship is always searching for this very large whale called Moby Dick. He spends vast amounts of resources trying to find the whale. And so this is what I mean by white whales, this kind of a tongue-in-cheek thing, because he did actually make some progress on this, but it was something that took years and years and years. And of course, in science, sometimes it does take years and years and years to devote yourself to ideas and to shape them. And sometimes you never see sort of the payoff in your lifetime, but you end up doing it and you end up advancing knowledge. And that's kind of the important part. So when we talk about differentiation waves, we're talking about different types of like collective behaviors of cells. So this is an example of a calcium wave in development. And this is, of course, the time series for this calcium wave. So there's this calcium wave that gets activated, and then this primed component here. So we have this translocation phase, the diffusion activation phase, and then the wave sets itself down. So this is where we see embryonic waves in Xenopus in different phases of development. From fertilization, there's a fertilization wave, there's a post-fertilization wave, there's surface contraction waves, there's a first division or cleavage, then there's FGF induction, another cell division wave, and some other things. So he saw these waves and built a theory around it, which is, a nice sort of way to summarize theoretically a lot of these wave-like behaviors that were going on in embryos. And so, you have these waves that are defined in the literature, and he kind of brought that all together under this idea of differentiation waves and making predictions about how those might guide development at a higher, um, at greater time scales than just kind of these localized events. Now, what we were interested in DevoWorm was we were interested, of course, in C. elegans, but we did spend some time investigating this problem in Drosophila, which is a fruit fly. And so Drosophila embryos have very interesting properties. We have these cellularized embryos here on the right that exhibit mitotic waves. So that means that you have waves that are concurrent with cell division. We also have these imaginal discs, which form the precursors of the eye and the precursors of the wing. And we can see this in the development of the eye, where there's this furrow that moves across the imaginal disc and results in different types of eye morphogenesis. So we see differences in mutants. We see differences in how the furrow unfolds across that imaginal disc. And then we see differences in the adult phenotype. So we'll get to that work in a bit. But these are some screenshots from his book. We have this figure that he proposed. So he developed this theory of differentiation waves, but then he mapped this to trees and codes. And this is an example of a differentiation tree. So the idea is those waves are set up and they move across the tissue. And this guides different differentiation events. So you could have cells in an imaginal disc, which is undifferentiated, and those waves move across that tissue and result in cell division events or cell differentiation events. And those cell differentiation events can be marked by some sort of bifurcation of a cell lineage. So that bifurcation then are branches on a tree. And so these are some shots from the book. The book is very theoretical in the sense that it has a lot of diagrams, it has a lot of ideas, but some of it is mapped to the biology and some of it is just kind of this should be what we should expect. Different types of predictions that aren't shown in like an actual biological system. Nevertheless, he gives an example here from metamorphosis, which you see in a lot of insects, where they start life as in one morphological configuration, and then they go into like a cocoon, and then they come out as a different configuration. So if we think about caterpillars turning into butterflies, the caterpillar is in a certain a certain morphological configuration at one stage of its life. It goes into a cocoon where a lot of the cells are a lot of the tissues are decellularized is the terminology, and then those tissues are rebuilt with different cell differentiation events, and then you end up with a butterfly emerging from the cocoon. So the cocoon is the site of a lot of dedifferentiation, and so we see that here where we have this tree that leads from an embryo, a fertilized embryo, to the caterpillar phenotype. And then, of course, we have this set of decellularization events where things just kind of dissolve. We get a single lineage that persists throughout metamorphosis. And then we get these differentiation events leading to the caterpillar phenotype down here. So this is just one example. I think he talks about the Ascidian larva, which is, you can evaluate these different morphologies in terms of their phylogenetic traits and things like that. And it's an interesting problem because it poses this problem of the differentiation trees being this unfolding set of differentiation events. But then what happens when that's reversed? So sometimes in regeneration, like in some fishes and in some other organisms, you get tail regeneration, where the site of the wound, you get this dedifferentiation of cells and you get this growth, and then you get a new tail. And it can also be described roughly by this kind of diagram, where you have a bunch of branching events. Those branches terminate. There's a single branch that continues, And then from that is the source of all the diversity of the new phenotype. On the right hand side here, this is just showing differentiation trees in space, space of possibilities for different types of differentiation trees. So in this case, we have um, different differentiation trees of different complexity. So the more branches you have, the greater the complexity of the differentiation tree, the more differentiation events you have. Perhaps that means you have a longer developmental time, or perhaps that means you have a more complex phenotype. And so this figure is organized radially by complexity, so you can see that with evolutionary time, the prediction is that these differentiation trees will grow in complexity. And this is something that is predicted also by the wall of complexity that Stephen Jay Gould proposed about how we have sort of this progressive increase in complexity and evolution over time. So we worked out a differentiation tree for what's called mosaic development. So mosaic development is something you see in ascidians early on, which are sea slugs. So it's a marine invertebrate. And then you also see it in C. elegans throughout the lineage tree. For the most part, there's some parts that are what they call regulative development, where they're not the cell divisions are not deterministic, the cell differentiations aren't deterministic. So that's a difference. So mosaic development is where you have these deterministic divisions, these deterministic differentiation events, meaning that you have a cell lineage that unfolds on a regular basis across individuals. So there's a timing that's persistent across individuals. But you also have this sort of program nature of differentiation. In other words, differentiation events will always happen at a certain point in developmental time. You can't really push those around without disrupting development. Whereas in regulative development, it really depends on the signals in the environment more, and individual cells can kind of have different identities depending on how the embryo is configured. So in C. elegans, the developmental process is very much unfolds in a very predictable way. We can identify cells, we can identify cell division, and we have like a nomenclature for all of that. We have a lineage tree that we can build that is organized by AP axis, but it basically is these initial lineages sort of dividing into these different components of the phenotype. Now, this is an example of a differentiation tree. So we are taking what is known as the lineage tree in C. elegans, and we're using sort of the terminology of that lineage tree, but we're reorganizing it as a differentiation tree. So we start with a founder cell. And in C. elegans, we have 8 founder cells. They go from anterior to posterior, and the AB lineage is anterior. There are other founder cells in the posterior. And from those 8 cells, we get all of the tissues of the adult worm. So in the lineage tree, this is all organized by AP axis. So it's everything from the most anterior part to the most posterior part. That's the order. Now, in the differentiation tree, we have a different order, which is we will order these recursively by larger cell and smaller cell. So when the founder cell divides, the larger cell goes to the left, the smaller cell goes to the right. And there aren't huge differences in the cell size. The cell size decreases. So like the founder cell is a certain size. At the point of division, the 2 daughter cells are half the size of the founder cell, and they grow later, but like in terms of size, they get smaller and smaller. But there are differences between the 2 daughters in terms of size. And so the idea is that if there's an asymmetry in terms of size, that should have an effect in terms of the differentiation tree. If there are asymmetrical divisions, meaning that one daughter cell dies and doesn't divide again, which happens, that's another asymmetry. And these symmetry breaking events then lead to the transformation of a sphere or of a bean-shaped embryo to basically the highly asymmetrical phenotype that we see in the adult. And so this is the reordered tree, not by AP axis, but by size and fractions of size in different parts of the embryo. So there's this AP axial organization, but it's convoluted. And this can also represent local division and these expansion contraction waves operating locally. So this is what it's supposed to represent. By reorganizing it, we get like some sort of sense of what that looks like. So we wrote this paper, Quantifying Mosaic Development, back in 2016 that kind of laid this out for both C. elegans and for Ciona intestinalis, which is an ascidian. This is our differentiation tree for early Ciona intestinalis embryogenesis up to the 256 cell stage. And so this is where we have fate mapping that's worked out for this. But we also have the unfolding of this mosaic differentiation, or this mosaic lineage tree that was transformed into a differentiation tree. And so this is just a way of visualizing this, where we show like the division into founder cells. And, this is a fourfold symmetry. So it's like dividing into founder cells, and then it divides into these daughters down to the 256 cell stage. So it's saying that Ciona intestinalis embryos at that stage look very symmetrical, but they become asymmetrical later. And they have a different they have a compound development where they kind of form these very complex embryos early, and then they become like these tadpoles, and they're different phases of development. We published this work in our 2016 paper on quantifying mosaic development. But we also, this particular tree style, or this tree layout, is featured in this paper from 2023, Embodied Cognitive Morphogenesis, and that's in Royal Society Interface Focus. So these are differentiation waves, or actually rather, this is a high resolution diagram that someone actually drew by hand of the imaginal disc that will become an eye. And so what happens here is you have this furrow, which you can see here, and it moves across this imaginal disc from right to left. And then behind the furrow, you have these highly organized cellular structures. You have these nuclei of what they will call ommatidia in the eye. So the insect eye is different from the camera eye of the human, in that it has hundreds and hundreds of ommatidia surrounded by supporting cells. So you have these different ommatidia that are sort of these visual units. And then you have this furrow, which is a bunch of sort of cell differentiation going on. And then ahead of the furrow, you have these undifferentiated cells, where you can see that there are stem cells or other types of precursor cells sitting in there. So the idea is this furrow moves across, and as the furrow moves across, it leaves in its wake this differentiation. These differentiation events continue to the left-hand side of this disc. And so we can actually trace this wave across different parts of the imaginal disc. We did this using a high-resolution drawing of the third instar eye disc. So this is an example of this. And we have this disc that has the furrow here. And in this case, it's going to be moving from left to right. So we have this sort of shifted around, and we were looking at it in different geometries. And as we'll see, we can even project it as into different projections and look at that process in a conformal map. So this is a third instar eye disc. This was drawn by hand by Tanya Wolf, who was one of Dick's graduate students back in 1993. And then his mother, Diana Gordon, was an artist who touched up the image, closed some of the loops. So you have to appreciate that Tanya Wolf sat there at a microscope and drew all this out by hand. And this was not unlike what John Solston did with the C. elegans lineage tree. He sat by a microscope and drew that lineage tree out by hand. And so this is a very important but maybe unsung part of biology research, is being able to draw well and draw things out that you otherwise couldn't get at with imaging technologies. I know everything is about imaging technologies these days, but drawing things, it's not only a way to sort of bring some of these processes to life, but it also helps you learn, I think, a lot about biology, because if you have to draw out these things, especially as they sort of change over time, you get an appreciation for what they look like. You get an appreciation for the geometry, and you get an appreciation for the process. In any case, we published this paper in Biosystems in 2018. This is morphogenetic processes as data, quantitative structure in the Drosophila eye imaginal disc. And this is where we had just basically showing this theoretical cartoon here. So this just shows you what goes on in this just as a cross section of this third instar disk. And this is organized from left to right. So the furrow is moving right in this image. The furrow is identified here. The pre-furrow is identified here. So in the pre-furrow, you have these undifferentiated cells undergoing cell division. The furrow is where you get this contraction, so this contraction wave. And then you get these different clusters that are formed post-furrow. So you get this differentiation leading to these ommatidia, which you can see here. You start to see like different clusters, which are these R7 cells. You get cell divisions that lead to so you get the signal, the induction signal, and then you get these cell divisions, which lead to the final product of differentiated ommatidia. So that's what we're talking about here. So the furrow doesn't magically transform them into ommatidia. It actually induces a series of events which lead, so you get some transformations like differentiation, then you get cell divisions, and then you end up with these structures. That's something that we can extract from like an image, from a microscopy image. This is the high resolution drawing showing all the cells. And then this is where we segmented this drawing and found the cell centroid position. So we were able to characterize each cell by its spatial location in this image, and work with it from there. So we had this point cloud, or this point array of the third star eye disk. And so this is an example of the segmented image. This shows the ommatidia pulled out of the segmentation process. So this is what structured tissue looks like in terms of the ommatidia. All those white dots are cell centroids. And so this is just showing this pattern, and then showing we took out the furrow here and the pre-furrow. So this just shows the structured tissue. And then this just shows kind of what we think is going on. So, the idea here is that you actually have a combination of expansion and contraction waves happening with the furrow, but also happening behind the furrow. So the contraction wave is with the furrow, and then the expansion wave is behind the furrow, leading to these cell division events that lead to these ommatidia. And so we're able to take this. This is an example on the far left-hand side of the slide of the third instar imaginal disk, sort of a high resolution microscopy image. And then we have this drawing here, which is the high resolution drawing. Then we have the segmentation map, which is basically taking the cell centroids out and then straightening out the imaginal disc so that it is not curved, so it's straight along the furrow. And this is as straight as we could get it. And so we have our sheet, and so we can observe that process in a linear fashion. But then we also made a conformal map where we showed kind of the structure in this projection, which is a spherical projection. So this is an example of computational developmental biology, taking biological data and transforming it into things that can be computed on. So you might ask, like, what's going on after the furrow passes by these cells? I mean, it sends a signal, and then there's this induction of something in the cells, and it leads to this differentiation. Well, what Dick hypothesized basically is that there is some sort of mechanism in the cell that responds to the signals from those waves that coordinates the behaviors of cells behind the wave. And so his idea was there was a cell state splitter mechanism. And what he did was he had this idea of a mechanism, and he mapped it to different things within the cell that might be plausible sites of action. So this is a figure. This is I can't remember if this is from one of his papers on the cell state splitter or if this is from the book. But basically, this shows the cytoskeleton and how the cytoskeleton is organized and how that could be the site of the cell state splitter. So the idea is you get the signal, it induces a change in the cell. It's usually some sort of differentiation event. So it changes the morphology of the cell and it changes the gene expression of the cell. And that all happens by reconfiguring the cytoskeleton first and then sending other signals down to the nucleus and to other parts of the cell morphology. And there's a feedback there that leads to these differentiation events. So this is an example of what he thinks is going on in the cytoskeleton. You have this microfilament ring, you have these apical microtubules, you have these other things in membrane that are membrane bound, and then you have these intermediate filaments and paraxial microtubules. So there are a lot of things going on in the cytoskeleton, which is basically the skeleton of the cell. And it's sort of underneath the membrane, so it gives the cell its rigidity. And so the idea is that there are changes going on here that will lead to this sort of differentiation event. And, it's not exactly clear about the causality, but the idea being these are the key components. So you can also have a cell state splitter that's activated in one cell, and then that triggers a contraction wave in the neighboring cells, which is interesting because it's not just about getting signals from the environment, it's about interactions between the environment, the cell, one cell changing shape, and then that propagating across the population of cells. Because remember, tissues are populations of cells. The example we saw before of the imaginal disc, that's a population of cells. So this is just a cartoon showing that you have this cell state splitter activated, triggering a contraction wave. In this case, a cell state splitter is activated and it triggers an expansion wave. So you can have both types of waves, expansion and contraction, due to cell state splitter activation. And again, it's just that you activate things in one cell, it propagates to the next cell, it propagates to the next cell, and you get these waves across the embryo where you get cell by cell by cell going across. This is a set of diagrams from a paper we published maybe only a few years ago, in 2024, and this was in the journal Biosystems. And this was an attempt to sort of connect molecular components like proteins and transcription factors to this process of differentiation wave activity. So this was a very complex paper to work on. We had 4 model organisms where we looked at proteins and transcription factor activity in the development of these 4 model organisms, looking at secondary data from PubMed, downloading them, doing the analysis, and then mapping them to development and perhaps even the morphology. So we didn't really get very far in the morphology part in this paper, but we do map this to the developmental time course and then identify key transcription factors and proteins that are involved in this. So this is a map of C. elegans here where we have the surface going from mid to early development. So we had a couple of data sets that we aligned and then looked at the activity of these different developmental candidates. So we have a lot of different genes in C. elegans that are involved in these different encoding these different proteins that are shown in the brackets. And so we see that, like, we get see Wnt involvement, rack involvement, MAPK, GSK, APC. And this is the expression level. So we get the expression level of the uh, transcription factors, the associated protein. We also have the protein counts. So we can say that there's this correspondence between the protein and the mRNA, although technically that's a very poor correlation. But we can actually get candidates and look at them. Um, and so this is a nice little map of this in C. elegans. And I'm showing the Drosophila ommatidia here just as an example of what we're thinking. So these are cells that have undergone this process of differentiation, a differentiation wave going across a tissue, and then you get these signatures as you go through development. So as you start here, you should see maybe less, our surface kind of undulates upward here, not by a lot, but by some, because remember, you still have developmental things going on here. And so this is just a nice sort of first pass approximation at this sort of project. So we'll put some sort of modern molecular and cell biology skin onto the skeleton of a theory. Okay, so that's, and he published a lot on differentiation waves and differentiation trees, and we did a lot of that in DevoWorm in our collaboration, and he's not, with us anymore. He really didn't pursue that, I don't think, after 2024 much, because he was working on his embryo physics stuff, and that was very important to finish for him because he knew his years were short. So it might be interesting to pick that up where he left off and continue on with some of that. That book was extremely large, voluminous, thousands of, probably over 1,000 pages, and it may be even 1,500 pages. Some of that were citations, of course, but there was a lot of stuff in that book that could be explored by someone who really wanted to put some of these theories to the test or go back and look at what he was actually talking about. So this is his diatom work. So I say diatoms are everything because diatoms actually are model organisms, but they're also used in things like biofuels. And they're very interesting organisms. They're an algae, but they have these glass membranes. So they have these distinct shapes where you have stars and circles and these long rods and other types of things that form. They're very rigid membranes. And, they also can behave collectively. They can also do other things as well. So they're a very diverse set of model organisms. They're relatively easy to raise in culture and relatively easy to do all sorts of different types of behavioral experiments and other types of experiments as well, metabolic experiments, etc. Dick worked on a number of papers in this area, but also a number of books. This is a book called Diatom Gliding Motility. So this is edited by Stanley Cohen and Kalina Manoilov and Richard Gordon. So, this was something that we contributed a paper to. But the idea was he's interested in this one of the questions in diatoms, and he was interested in a lot of different questions relative to diatoms. But one of his main questions that he wanted to answer was, if you look at this colony here, how does this colony move across the water column? And so you find diatoms in these very shallow aquatic environments. So they're in an aquatic environment. They have access to sunlight, and they move through the water column. They respond to sunlight. They respond to salinity gradients, and they move according to this sort of gliding mechanism. And we'll get to that in a minute. That's what our paper was actually about directly. But if we look on the left-hand side of the slide, we see that there are different shapes of diatoms. They're actually very small organisms. They're 10 to 80 microns in size, depending on the species and the phenotype. This is a raphid diatom, which is a type of diatom that is rod-shaped. So there's the frustral, the raphe, the epivalve, the hypovalve. So you have these girdle bands. And if you look at this picture on the right, you see that this is a raphe diatom with a number of these different rod-shaped diatoms aligned in parallel. And they move against one another in this gliding motion that defines the colony. So sometimes they live in colonies, sometimes they live solitary. And this is just one cell. So this is the cell itself. And remember, they have this hard um, silicate membrane. So it's not like we have a very soft membrane like we had with C. elegans or with Drosophila. And we have these different shapes. So we have rod-shaped diatoms, flake-shaped diatoms, and then these three-dimensional shapes, which is really interesting because they almost look like they're 3D printed, but they're not. They're actual. They're organized, very small organized phenotypes. So it's very interesting to look at biocomplexity in diatoms. This is one species or one genera of rod-shaped gliding diatoms. This is Basilaria spa. So spa just means it's a number of different species that we can't define their differences, but we just classify it under this genus Basilaria. In any case, we have these rod-shaped diatoms that live in parallel in these colonies, and you see that they are sort of stuck together along the edge. So they actually move according to this accordion type movement. So they kind of glide apart and then glide together. It's like playing an accordion. You move each end of the colony apart and then push it back together, and then that's how they move across the water column. You can actually see there's a lot of like high resolution electron microscopy of these organisms. So you know that they're very small. They're on the micron scale, but you have this this phenotype that you can see the high levels of detail on. And this is this is another one that's a little bit bigger than this is a 10 micron bar. So this is actually in the hundreds of microns, or at least I guess maybe 100 microns. But basically, that's our size scale, and then we can zoom in and see these features of the phenotype. So it's actually quite complex what's going on here with our diatom colonies. And it means that we can understand their movement. So we can understand maybe what the mechanisms are for the movement, how they're moving, and so forth. Now we can do a mathematical analysis of their movements. So remember I said that they glide and they oscillate according to this sort of accordion model where you pull it apart, pull the colony apart, push the colony back together, and it does that to move. Now this is a phase portrait of these oscillations. So we can take video of these movements of these colonies, and we can take the images and segment them like we did with our Drosophila work. And we can actually identify centroids or other features of each cell. And then we can plot those in a spatial representation where we just measure the distance between the centroids or distance between the edges of the cell. And we can get these measurements that then we can analyze. And we can use these phase portraits to characterize the oscillation as an attractor basin, as a circular plot here. So this is where we have position versus velocity, position in microns, velocity in microns per second. And we see that, as the movement of this accordion reaches its apex, it's extended in terms of its position, and then it collapses as the velocity increases positively or negatively. So as it's kind of moving outward, it reaches the full extent of its position from the leading cell to the lagging cell at the end of the colony. And then as it's pushing back together, it's either slowing down or speeding up. So that's what this describes. And then you see this noise along each part of this. So as it's speeding up or as it's slowing down, it's got this noise associated with it. And so we can actually describe this oscillator as a delayed harmonic oscillator. So this oscillator, as you can see, it's very similar over time. It's just that there's some noise in here which we can capture with this harmonic oscillator. So I captured this first order approximation of the harmonic oscillator, and this is the mathematical description. So we can model this in a modeling software. One of the interesting parts of this is that we see these noise periods here. And this is, we know from sort of observing the system that that's the end of the oscillation. And so we have this debate about, like, what does this represent? What is this? So if we know anything about kinematics, we know that when you get a signal like this where there's a high degree of variation across sort of the derivatives of motion, it's a higher order of motion. So you get things like jerkiness, where you get these higher order approximations called jerk. So we can call it jerkiness. And this just means that there's a lot of variation in the acceleration during this time. And so this is the third derivative of position jerk, which is, I think, what we're capturing here. It's not really clear, but that's probably what it is. So it's interesting to see these different kinematic components emerge out of this analysis. So then, we published a paper, actually 2 papers on that. The other paper was on characterizing the behaviors of diatoms using sort of a psychophysical framework, which is interesting because, as I said, diatoms respond to light, and they respond to salinity gradients and other types of chemical stimuli. And so thinking about this problem in terms of those stimuli are very interesting as well. And we published a paper on that. He also was, of course, interested in origins of life. And so his origins of life work were maybe less prolific than the other things, but it's worth talking about. We published a paper in 2024, which was called An Ensemble Approach to the Origin of Life. I'm going to be presenting on this work at the Artificial Light Conference this year. So if you're interested, it's in one of the workshops. So I'll be posting a video on that closer to the conference on the DevoWorm YouTube channel if you're interested. So one of the things about our work in this book, Origin of Life via Alicea, and Dick published some other work in this book, including looking at shaped droplets of different shapes, and he published that with an alum of the DevoWorm group, Mayuk Deb, and so that's also in this book. But I want to talk about this ensemble approach to early life, or the ensemble approach to the origin of life paper that we published as chapter 8. And so the interest here in early life was in these early life ensembles and how they lead to pattern formation. So an ensemble is something, we've got to borrow from information theory, where you have components of different types in an ensemble, which would be like a population of things or a collection of things. And the ensemble changes based on the frequency of these different states in that collection of things. So if you have a well-mixed collection of things, half of them being black dots, half of them being white dots, that's what we might call maximum entropy, or a well-mixed ensemble. If there are more black than white dots or more white than black dots, then there's a bias in there, and there's less information in that ensemble, and it's easier to predict what that ensemble contains. So just a little bit of shorthand about this. And then, of course, leading to pattern formation, then we can measure out, based on the state of the ensemble, when pattern formation occurs and what's starting, and then how it proceeds. So we looked at 2 questions here in this paper. We used these different computational models for each. So the first was origins of evolvability. We basically asked the question, can we construct sustained biocomplexity or evolve some sort of phylogeny from what we might call a random parts list? So if we had a bunch of components sitting around a bunch of biomolecules or other pieces, can we assemble some sort of biocomplexity that evolves, or that at least replicates in a sustained fashion. And we did some experiments with these vehicles. They're kind of inspired by Bratenberg vehicles, but they're basically this parts list that get put together, and then you get these lineages that emerge spontaneously. So the answer to that is we could, and it actually is quite common if you have a parts list that can be then, um, in a situation where it can be put together or self-assembled. So that was one example. And then the other example is the role of compartmentalization. So a lot of early life experiments involve some sort of vesicle and booting up a vesicle with biomolecules, and then those biomolecules being in a contained space can perform reactions and form this sort of sustained complex system. And so we built these models, these computational models of different vesicles with pores, and the pores could close or open as we saw fit, and we kind of mapped that to day-night cycles in the simulation, so that you get these compartments that will be havens for reactivity, and then you get complexity emerging within a cell that way. And so our vesicles are a container for biomolecular reactions. And these are some graphs from the paper where we kind of worked out some examples of, how many molecules do you get when your container is of a certain size, which means if you have a larger container, you get more types of molecules, and the ensemble is greater. And then here you have the net molecules. So if you get flux, where things are moving in and out of the membrane, actually I think it's -1. So low levels of flux show the sort of low number of net molecules. High levels of flux show this greater level of molecules. And I'm not sure why the numbers are like that, but I think it's because we did a 1- transformation on that. So it's actually, this is a low level of flux, this is a high level of flux. In any case, what we're doing is we're blocking off these membrane pores, and we're opening them back up. And so that's what this represents. And then the net molecular content is where you measure how many molecules you have over time. And you can see that you have a greater number for this level of flux than this level of flux. Okay, so then I didn't really put in this part of it, but we actually looked at day-night cycles. So when you vary the day-night cycle, if you have a longer day-night oscillation, so if you have longer days and longer nights, then this process slows down. Whereas if you have shorter days and shorter nights, it speeds up. And this just speaks to like astrobiology. If you had a planet where the day-night cycles are very quick versus where the day-night cycles are very long, and it depends on how the planet is spinning on its axis. So if it's doing it really slowly, then you have this different, a different probability of life occurring than if it's moving very quickly on its axis. So given that work, Dick always loved posing open questions. So he would spend time coming up with all these questions about things as he read things, and he read a lot of things. And he would always put these open questions out for people to answer, students, other researchers, and many more questions that could be answered. But it was all about getting people to think about things and to latch onto something sort of collaborate on, or maybe improve their understanding of something. So these open questions are pedagogical, but they're also about collaborating. This is one example of some open questions that he asked. This was circa December 2023. So in December 2023, I think in our last DevoWorm meeting of the year, he posed these open questions. So they were just really things that you have to unpack, and you have to maybe do some background reading and some of them are actually quite useful. Some of them maybe less so. So this, we were talking at the time about networks, and how we could have embryo networks where each cell formed a node in the network. And we were talking about how those networks evolved over time or changed their configuration. And he had this idea of how we could look at the changing network of neighbors from the point of view of one cell in an embryo. So using one cell as sort of the reference frame, what does it look like to be in that cell, and what neighbors are near you, and what does it mean in terms of signaling, in terms of cell migration and other things like that. It seems far out at first, but then you start to think about it. Oh yes, this makes sense. Then another question was, can we shift diatoms, in this case cells, it could be generalized cells, from one category to another with something like a compustat, which is this, it's like a chemostat, but like using a computer. We were talking about a compustat at the time, so this is kind of an internal reference. Then he had some points about the early development of caecilians or apoda. You see an example of this here. There are these worms that are that lay eggs in these clutches, and this is just something he was interested in because he was reading about it. Review of the literature with the translation of Saracen and Saracen. This was a 19th century paper from 1889 called On the Developmental History and Anatomy of the Ceylon Blind Burrower, Ichthyophis glutinosus. So this is the organism in question. So, he loved to dig back into the literature quite a bit, like, into the literature of the 19th century and the early 20th century, going to very obscure papers and trying to pull things out and bring them to life. So one of his other open questions, he actually published a paper on biosystems in 2025 on this idea of working on different aspects of archaea. So this paper was a call for research on the basis for polygonal pleomorphism in archaea. Like I said, he published that paper on different cell shapes in archaea, like triangles and circles and stars and things like that. And archaea has these different shaped cells. And the question is, how do they form? And what he calls polygonal pleomorphism, which are changes in the shape of the polygon. And so this actually is, this paper, if you go to it, it's actually 20 open questions for astrobiology. Because there is this link between archaeobacteria and astrobiology, and that archaeobacteria represent, a lot of times they represent examples of organisms that evolve in extreme environments on Earth. And we know that there are a lot of planets that have what we might find equivalent to extreme environments on Earth. And so the question is, maybe archaea tell us a lot about evolution on other planets than maybe like plants or animals or other bacteria. So these are 20 open questions for astrobiology, and you can go to the paper to see what those are. But he talks about this is his sort of summary. Investigation of these and related questions about polygonal archaea could be quite fruitful and have implications for multicellular organisms. While much of the archaea literature is taxonomic, meaning that you look at like the different things that define different species or different lineages of archaea, this domain deserves much more cell biology investigation. So archaea cell biology is not as up to speed as the stuff that you see on taxonomy. Its role in the taxonomic order, like, looking at the diversity of cells and things like that. There's more to archaea than just that. There's a lot of cell biology. There's also a lot of molecular biology in archaea, but not linking that up with the cell biology. And that's, of course, important for astrobiology. So it's really, again, these questions are to get you thinking about things in a certain domain. It's not meant to be like a refined set of research questions. So another idea that we had that kind of made it into another book, and then maybe is the precursor for additional work, is this idea where he proposed an idea once in DevoWorm where we kind of had this open question or this open thing that he wanted to find out more about. And it was that, can we use Perlin noise, which is a form of procedurally generated noise, as a way to mask tumors in an image? So, tumors are hard to find in biomedical images. Oftentimes they're masked by their tissues. They're kind of hidden by maybe like noise in the image. And so the idea is, can we simulate that noise and simulate it in a way that allows us to see sort of the resolution of a certain imaging or computational technique to find tumors. We know that there's a tumor in the image, we just mask it with Perlin noise, and we can find it underneath, given that we can tune Perlin noise and that we know that what the noiseless image looks like. So then that was the question. And so in the next meeting, I prepared a Python interactive notebook where I did a Perlin noise demo. I worked out some problems. I showed how it works, how we could modify it, how we could mask things using Perlin noise. And so you can do this in Python. And so Dick wanted to publish a book chapter on it. We published a book chapter, and it kind of evolved a little bit from the original demo to this idea of contrast to Perlin noise and avoiding it. So this is where he picked up on this idea that Perlin noise might be used as an analog for tissue. So again, we could use it as just pure noise or some sort of procedurally generated noise, meaning that we don't want to use like kind of Gaussian noise, because Gaussian noise, we kind of know what that does. We know what that mask looks like. We want to mimic something that is sort of heterogeneous. So Perlin noise it is. And so this is where he picked up on this idea of using it as an analog for different tissues. So this ended up in this book, Steerable Ray by Ray X-ray Computed Tomography, A Plan to End Breast Cancer. This isn't out yet. This is the last thing he worked on in his life. And he worked on it with Syed Hussein Ather, who is actually someone I began mentoring in DevoWorm, and then he worked with Dick on this work. And he's really come out of his shell in terms of being a collaborator because of our interactions. But we will be publishing this chapter 11 in the book. And then that's also led me, one of the last messages I received from Dick was saying, you should pursue this idea. So I've been pursuing it since. And I've been working out, using agentive coding, ways to like model tissues using Perlin noise. So not just using it as a mask, but actually using Perlin noise to generate different tissue types, or using an evolutionary algorithm or something similar to evolve from Perlin noise, from pure Perlin noise to different tissue types or things that mimic different tissue types, and figuring out what the parameters are for that. So that's a work in progress, but it just goes to show you how these open questions can evolve into really fruitful areas of research. This is an example of what I mean. This is, again, some agentive coding where we go from like a standard sheet of Perlin noise to these different, this is an animation in the middle. These are static images on the sides, but showing that they're different sort of tissues that can be modeled using Perlin noise. So this is Perlin noise as a synthetic lobular tissue. Perlin noise is an animated myocardial volume, so you can see it looks like striated heart muscle. And then this is Perlin noise as a myocardial sheet. So it's a little grainy, but you can see that it's a little bit different than this one. I'm continuing to work on this, but I think it's really fruitful. It looks interesting, and let's stay tuned for that. So yeah, in terms of mentorship, Dick was a wonderful mentor. He was a mentor to me and many other people. A lot of DevoWorm. We, as our collaboration developed, we kind of traded mentees where they kind of moved between us. And it was interesting because we had people, start with Dick or start with myself and then move on to like another person depending on their interests. And DevoWorm became a great catalyst for this. So he really did mentor a lot of scholars all over the world. He did a lot of mentorship via the internet, through digital means, and it was I don't know how many mentees he had, probably in the thousands, at least hundreds. I know that. So there were a couple of people who I think deserve mention here. Hussein Ather, who started out working with me in a Google Summer of Code project, and then is interested in cognitive science, and we worked on some cognitive science things. And then he also attended DevoWorm, And he eventually started working with Dick on this cancer physics idea because he had a background in like philosophy and physics, very eclectic background. And so he's been in, you know, this is an example of someone who really started out really unsure of himself professionally. And he had some mistakes that he made in his professional life and all this. And working with us, I think he really kind of became much more effective as an academic, as a researcher, and you know, I like to think our mentorship helped a lot. Rob Stone, who is sort of an independent researcher, he started out working with Dick on this idea of cybernetics and the cybernetic embryo. And then we started collaborating on thinking about questions in cybernetics as well. Then Susan Crawford Young, who's a regular at DevoWorm, she was one of Dick's students, also at the University of Manitoba. And then through DevoWorm, we started collaborating on some topics in the meetings. So interesting things like the different aspects of embryo physics related to like mechanical stress and strain and tensegrity, which is a type of ultra stability. So and then of course, I think her interests have expanded on to other topics, like in terms of networks or other things that we mentioned in the meeting. And, I think this has been very good for her professional development as well. This, speaking of Hussein Ather, he presented a LinkedIn tribute to Dick right after he died. So this was his LinkedIn tribute. I showed a obituary from his local paper in the first slide, but this is Hussein's kind of professional um, obituary here. And it was just his own experience and, some comments and things like that. So that was a nice thing to kind of, and they don't have the whole message here, but basically, it's good to see that, like, when you die, you have a legacy. And Dick and I talked about legacy like a year before he died, and thinking not only about who you influence and what you put out into the world and how that lasts and persists, but also about what happens to your stuff when you die. Do you archive it? Do people see it after you die? How do you organize your things? It's almost like a last academic or last researcher will and testament. And so this is an important thing I think to think about. We didn't have any clear answers, so I don't know what the plans are for his stuff, but I will. We'll see. But I think he's left a pretty good legacy.